First of all, I just want to explain what this presentation is all about. It is meant for new incoming students in my group that have some background in engineering, very little background in biology, and don't know what cryobiology is. Just to be sure that it is understood. This is not a seminar that is going to present the latest in the field or any huge breakthroughs in my lab. It is meant for new incoming graduate students in engineering with a background in engineering, who know what heat transfer is, and who really don't know very much about biology, probably don't know very much about cryobiology, who might want to confess that it's more working in cryobiology. So, the research in this field, the primary motivation in the field is organ transplant. Essentially, a large number of organ transplants are available but are not being used because of the lack of means to preserve those organs, because of technical difficulties with the transplanted organs. So the hope is to find ways to preserve organs for extended period of time to facilitate transplantation. And that's relevant to organs from donors, and it's relevant to organs done by tissue engineering, and it's relevant essentially to any other means to get an organ. So it's not only for organs from donors. As you can see here in this presentation, the number of people waiting for organ transplant and the number of people who get organ transplants, there's a huge difference. And those that do not get organ transplant are done. There are several techniques for organ preservation, but the technology that I will discuss is using subnormal thermic temperatures, meaning not the normal temperatures of life, but rather temperatures below the normal temperature of life. So the question is, why use subnormal thermic temperatures? And I will try to explain to you why to use those temperatures, what are the problems with those temperatures, and how to overcome the problems with those temperatures. So let me start with some facts. Again, for the engineers among us, the average temperature on Earth is about 50 degrees centigrade. At the end of the slide, you'll know why, and why the increase in global temperature probably causes essentially a change in the genetics of our species. The range of temperatures on Earth are between 70 degrees and minus 82 degrees or so. So that's the range of temperatures that exist on Earth. Also relevant to that, life has evolved to suit the environmental conditions in which it exists. Okay, so life has evolved with respect to these 50 degrees centigrade temperature, average temperature. It has also evolved to see in the peak frequency that the sun radiates. Okay, so it's important to realize that living organisms adjust themselves to their environment, or have evolved to adjust themselves to their environment. And if their environment changes, they have to readjust. It's possible, but they will not be the same. So this is another fact, that at atmospheric pressure, water freezes at 0 degrees centigrade. This is a fact that we will discuss later. Then life is really governed by the second law of thermodynamics. I will not dwell on the definition of the second law of thermodynamics in this point, but rather what it means. It means that heat has to go, can go, only from a high temperature to a low temperature. It cannot go in reverse. And when it does, it increases the entropy of the universe, essentially. So life has evolved to satisfy the second law of thermodynamics, which involves heat transfer from a living organism into the environmental temperature at 50 degrees centigrade. And that's relevant to normothermic conditions. This is my philosophy. So life is an electrochemical reaction, and the ultimate outcome is the increase of the entropy of the universe. So when the entropy of the universe gets to maximum, the Messiah will come. That's what life does, but essentially, in a more simplistic way, for life to occur, heat must flow from the system to the surroundings. Now, different organisms exist in nature that survive at different temperatures. So there are organisms or archaea that can grow up to 122 degrees centigrade. So life is not restricted to 36.7 degrees centigrade like that of mammals. So it can happen at higher temperature. It can happen essentially up to a temperature of 60 degrees centigrade, but most organisms cannot function anymore at 40 degrees centigrade. So that's one limit that we experience. The lower limit, there are organisms that can survive up to minus 20 degrees centigrade, and some of them can even survive down to minus 70. There are some insects that survive to minus 70 degrees centigrade, but life processes appear to be limited to minus 2 degrees centigrade. And this is the famous Arthur Clarke, the science fiction writer who invented the telecommunication satellites idea. So that's his review. So what I want to discuss now, what is happening to cells and the effects of subnormal thermic temperatures of cells from organisms that have evolved to live at 37 degrees centigrade. So there are two elements that are affected by these subnormal thermic temperatures. One is the cell membrane, and the second is the electrochemical reactions that occur inside the cell. And again, for those who have not seen any biology, life processes in a cell are essentially a process in which food is taken into the cell, it is decomposed to the basic elements, and as part of the process, heat is released. And that's the crucial element. When heat is released, you need a surrounding that is at a lower temperature than the cell. If it's not, then the cell cannot survive. And another element is that it generates energy in a chemical form, and this energy is used to make the new protein, the new structures, the new molecules that are suitable for the life processes. So this is in a nutshell what's happening in life. There is a need to balance the decomposition of food intake and the assembly of the desired molecules. To make it efficiently, and because these processes occur at a constant temperature, so organisms exist at a constant temperature, Now, chemical reactions are temperature dependent. To make possible for the composition or decomposition of molecules, there are certain proteins known as enzymes, and that's important for subnormothermic preservation. These enzymes essentially make possible the assembly of the new molecules and the disassembly of the old molecules at the optimal temperature at which cells survive or function. So this is the relation to preservation. Normally, life processes require enzymes for chemical reactions to occur, and these enzymes are optimized for a certain temperature. So, what is happening when you reduce the temperature? First of all, the entire process described here is known as the metabolism. So you probably have heard of metabolism. It's the entirety of chemical reactions that occur in cells. And the metabolism is a function of temperature. And there is a very important relation between the temperature and the rate of chemical reactions in living organisms, which is described here. Okay? And this reaction tells you that if you are at a certain temperature, R1, and then you move to another temperature, T2, there is a relation with a quantity known as Q10. Could I do something? So it's a relation known as Q10. I should keep an eye here. It tells me not to go to the point. So this relation tells you how the metabolism changes with temperature. And one of the key elements in subnormothermic preservation is that you want to reduce the metabolism so that undesirable chemical reactions cannot occur. So if you, now typical values for Q10 are between 2 and 3 for biological organs. And if you look at, if you decrease the temperature by 10 degrees centigrade, you can see that you can reduce the metabolism by a factor of 3 to the, or 2 or 3. Okay, so you can, by just reducing the temperature by 10 degrees centigrade, you substantially reduce the metabolism, and then you reduce the rate of chemical reactions. And in every paper that we write about preservation, we're using this equation. Now, this is a typical behavior. So obviously, as you of enzymes, as you increase the temperature, the rate of the chemical reactions increases until you get to a peak at which essentially denaturation of proteins occur. They completely lose their structure. So actually, this type of U-shape phenomena is quite typical in biological systems. Things go good, increase, and then they completely fall down. So that's what's the good effect of subnormothermic is. If you start from here, you reduce the temperature, the rate of chemical reactions is reduced, and essentially you don't generate, you don't use up the energy, you don't use up molecules, and in principle, the cell doesn't change any. And of course, if you go up, then you have protein denaturation. Now, these are enzymatic reactions, and they're different for different organisms. So, for instance, the organisms that can function at 100 degrees centigrade have different structure of enzymes. And that's what makes it possible. So the structure of the enzymes is important for a cell to function. But if you have organisms who peak enzyme activities about 39 degrees centigrade, if you go to higher temperatures, they will die. Okay, so we cannot survive temperatures above 42 degrees centigrade for substantial periods of time. Why? Because our enzymes kind of die. You reduce the temperature, you get to lower and lower enzymatic activity, and that's the subnormal thermal preservation. That's the good part of subnormal thermal preservation. So subnormal thermic temperatures reduce the rate of chemical reactions and reduce the deterioration of biological matter. So that's the good part. However, an organism has lots of elements, and there are bad parts. So now let's look at what's the detrimental effect of subnormal thermal temperatures on cells. One of them relates to proteins folding. Proteins must fold in order to function. They must take a particular shape. Now, taking the shape is related to the Gibbs free energy, but taking the shape is a kinetic phenomenon. And this here is the thing that's most important. The reaction, the kinetics of the reaction to get an optimal shape are temperature dependent. You can see here the temperature. So if you change the temperature substantially, then the proteins, meaning the enzymes, don't fold anymore in the shape that allows them to function as they would like to function. So that's a detrimental effect of reducing the temperature. Your enzymes begin to work in different ways that nature didn't meant it to work. So that's one bad aspect. The other bad aspect, which is even more important, has to do with the cell membrane. The cell membrane is a bilayer of lipid structures which has a polar head and a apolar tail. And the cell membrane looks like this, okay? A bilayer of lipids with proteins embedded in the cell membrane. So these lipids, what's happening to them when you reduce the temperature? These lipids affect the mass transfer between the interior and the exterior of the cell. As you reduce the temperature, these lipids become stiffer. And as they become stiffer, they don't function in terms of heat and mass transfer as they would in their normal state. And again, of course, this is saturation and unsaturation, which I will not go into the details, but the key element is that the fluidity of the lipid bilayer is essential for normal cell growth, and lower temperatures cause the lipid molecules to behave differently than their normal structure. And then essentially, as you go to very low temperatures, the cell membrane will lose structure. Just to give you a sense, when fat solidifies, that's essentially what's happening with lipids as you lower the temperature. So when the lipids their temperature is reduced below the optimal temperature, they lose their ability to control mass transport, and essentially, actually, they undergo phase separation, meaning that the molecules embedded in those membranes will essentially be compacted, moved along, even ejected from the cell membrane. So the cell membrane lipid structure is very important because it regulates the mass transfer between the interior of the cell and the exterior of the cell. And as you lower the temperature, let me just give you a sense. This is the effect of temperature on membrane permeability. And let's assume you have a 37 degrees centigrade organism. As you increase the temperature, the permeability increases, and again, water flows in and out, the cells dies. However, interestingly, as you lower the temperature, the permeability of the cell membrane at -5 degrees centigrade is comparable to the one at +5 degrees centigrade. So as you lower the temperature just to -5 degrees centigrade, the cell membrane becomes leaky. And that's a main issue because the interior of the cell is rich in potassium. The exterior is rich in sodium. And now when an uncontrolled mass transfer occurs across the cell membrane, when you reduce the temperature to below freezing, then the interior composition changes. And then when you warm up the tissue again, all the membrane proteins cannot function anymore, you lose calcium. So that's, I would say, that's one of the key elements. Most of the damage occurs not when you cool, but as you cool and then you warm up, the cell cannot function anymore because the interior environment is not proper for the cell to function. So actually, it was observed that cracks form in the cell membrane as you lower the temperature again, because there is a phase transformation. I will not go into the details, but lipids undergo a special phase transformation. And that's the key element. That's what people are addressing. How to avoid leakage of the cell membrane or how to handle the leakage of the cell membrane. Now, I mentioned to you that in nature there are organisms that live at -10 and +100. But how are they doing that? They actually change the lipid structure. So actually when bears undergo hibernation, they change the lipid structure of the cell membrane. So, everybody asks, well, bears can go into hibernation, organisms can go into sub-freezing temperature, but what's happening is that they change the lipid structure, and we cannot do that. So mammals, well, not all mammals, these mammals here cannot do that. And that's a main problem, and so unless we get our lipids changed somehow, we are unable to survive low temperature. So what are we going to do about that? So the main question is, how do you overcome the negative effects of subnormal temperature, knowing that the mass transfer across the cell membrane cannot be controlled anymore? And by the way, the cell membrane is probably the most important element of a cell, because it separates between the interior of the cell and the exterior of the cell. And if the cell membrane does not function properly, then the cell dies. And as evidence, there is no genetic disease related to the cell membrane, because every member of the population that has genetic modification in the cell membrane will die. So how are people solving that? They're solving the problem by changing the composition of the preservation solution. So one of the first steps was done by Southard and Belzer, University of Wisconsin solution, and they have replaced the extracellular solution in which the cells are placed
with an intracellular composition that is rich in potassium, so that there is no influx of sodium. But there are all these elements. Okay, so this is sodium and this is potassium. You can see solutions rich in potassium and then solution rich in sodium. And all these other elements that are needed for the cell to survive this excursion into low temperatures with changes in enzymes and changes in the interior composition. So the key element in subnormothermic preservation is the solution in which you keep the cells or the organism. Okay, so that's so right now organs for preservation are stored at low temperature. Their metabolism is being reduced. That's a good thing. The composition of the solutions is changed to adjust for membrane permeability. And right now, we can preserve organs at between 4 and -8 degrees centigrade. So that's great, but what's the problem? The problem is the time, okay? So at 4 degrees centigrade, you can preserve, for instance, a liver for 12 to 15 hours, a heart for 4 to 8 hours, and that's not enough for transplantation. It's not enough to take the organ from a donor in California and bring it to the East Coast. That's not enough to do eventually to 3D print an organ in California and bring it to the West Coast, or here in Berkeley to do a microchip that works with heart cells and bring them from the lab of Professor Healy here to a lab in, I don't know, in Minnesota. So that's a first task. How do we increase the preservation time? And one obvious solution is to reduce the temperature. Because if you reduce the temperature, if you remember, you have a Q between 2 and 3. If you reduce the temperature even further, then you can extend by a factor of 2 to 3 the preservation time. But of course, there is a problem with that. Now, I want to add some other things, and there are other elements. The elements are also optimal cooling and warming processes. The heat and mass transfer process is time dependent. Okay, so the mass transfer across the cell membrane is time dependent. The reorganization of the molecules is time dependent. So you can affect the outcome by cooling slower or cooling faster. You can affect the outcome by warming slower or warming faster, letting the system get used to the new configuration. Again, because it doesn't happen instantaneously. There is a strong time element. So this is something that we have shown also with the chip, with the heart chip, that the temperature, the rate at which you get to sub-freezing temperature is important. It will make a difference. So we have lots of parameters to study: the composition of the solution, the cooling rate, the warming rate, the temperature of preservation. Now there are a couple of new technologies that have been developed to reduce the temperature to sub-freezing temperature. And one of them is the supercooling. That's the group of Harvard, MIT, Mehmet, and Yarmush. They have developed techniques for stable supercooling. We have also used isochoric conditions, which I will discuss toward the end, to extend preservation. So what you want to do is, if you can, reduce the temperature of the biological organ or biological matter to sub-freezing temperatures and keep it there, you may have a chance to extend the preservation time, and experiments have demonstrated that's what's happening. So lower temperatures could further reduce the metabolism and extend the preservation time, but we have a problem. Water freezes at 0 degrees centigrade. And now we have another element that affects the survival of the cells, and that's the ice. And I will discuss in a few slides what does the ice do, but just briefly, Father Luyet was one of the leaders in cryopreservation, and the term cryobiology was introduced at that time, although cryobiology in biology doesn't mean cryogenics. It means below freezing temperatures. Cryogenic means minus the liquid gases. This is not liquid gases. So again, perhaps the biggest breakthrough in the field occurred when Paul, Smith, and Parks demonstrated that the addition of glycerol to sperm frozen to minus 79 degrees centigrade allowed the sperm to survive the freezing. That was essentially, it appears that other people have done that before, but this was a paper published in Nature and it took off from that time on. So this is the paper. It appeared in October 15, 1949, and this really started the new era of cryopreservation. So one of the most important researchers in the field of cryopreservation is Peter Maser. And Peter Maser wrote a fundamental paper in Science, I think it was in 1972, in which he described the effects of cooling rates on survival of cells to liquid nitrogen temperatures. And he has found the existence, or he described the existence of something that we call U-shaped survival curve. And in the U-shaped survival curve, you can see that as you increase the cooling rate, survival increases until you get to a point in which the survival decreases. So now the other interesting thing is that different cell types have different survival curves. And what we're going to do next is to try to understand how this happens as a means to develop new protocols. So freezing, you can look at the phase diagram. This is ice and water. If you are at a constant temperature, as you lower the temperature and you cross the so-called liquidus line between ice and water, everything freezes, undergoes phase transformation, and the liquid water becomes into an ice crystal. Now, this is for those of you who like mathematical modeling, and there are some, and I will not go into the details, but I just want to tell you that there are mathematical models that allow you to predict from heat transfer analysis the propagation of the change of phase interface inside the body. And in engineering, there are many people who spend all their career on developing mathematical modeling for heat transfer with phase transformation, because it's a nonlinear problem. So, and I will not go into the details of this, but again, it is possible to predict how the temperature evolves as you lower in a system, as you lower the outer surface temperature below the change of phase temperature, and how the change of phase interface propagates through ice into water. So you have here water, you have here ice. Okay. And I would say those of you that are interested in developing mathematical models for the process of cryopreservation, there is room for very interesting models to describe how fast it takes for an organ to freeze and what cooling rates occur. This is a microscope that we have developed that looks at a layer of water between two cover slips. And on that layer of water, we impose the temperature from a high temperature above freezing to a low temperature below freezing. And then we pushed the cover slip from the high temperature, we push the cover slip from the high temperature into the low temperature, and then a change of phase interface formed at 0 degrees centigrade. So this is how a change of phase interface looks under the microscope at 100 micron resolution. So you can see it's a straight line because it's a one-dimensional process. However, as you keep pushing the slide, you suddenly see that the interface changes form. So although the problem is still one-dimensional in heat transfer, a perturbation occurs on the change of phase interface, and dendritic spicular structures form. These are finger-like structures, and here you have ice crystals that form, and this is how actually water in biological system freezes. So when you freeze the water in biological systems, and I will explain shortly why this happens and how this happens. So what happens is ice has a tight crystallographic structure. Ice cannot contain any solutes. The biological fluid is essentially a fluid of saline and other components. So if freezing occurs, the solutes gather at the interface between the ice and water and increase the salinity. Now, phase transformation temperature depends on the concentration of salt. That's how we melt ice in the winter on the roads. So and by the way, these dots here, they are red blood cells. Same thing happens to the red blood cells. As we freeze, you can see here the salts and the red blood cells kind of gather at the interface. Now, this generates something known as a constitutive instability. I will not go into the details of it, but this instability is related to the fact that the diffusivity of salt, of mass, is much lower than the diffusivity of heat. So because the freezing interface piles up high concentrations of solutes, and the temperature goes down, you have regions in front of the change of phase interface that are thermodynamically supercooled. And because they're thermodynamically supercooled, and again, I'm just pointing you to what you may want to read later, but you should remember that there is an instability, and this instability causes the generation, if you have a perturbation, causes the generation of these dendrite ice crystal structures. And again, it's possible to develop mathematical models for that, and I've published on those, but again, I don't think you should, if you're interested, you can come later and we can discuss, but this is a field by itself. However, what's important is what's happening to cells when they're frozen. You can see here these red blood cells. The ice rejects the cells and it rejects the salt, generating a high concentration brine. And then these finger-like structures form, and these structures entrap between them the cells. Now, ice has no salt, has no solution. So now suddenly these cells find themselves in a high concentration salt solution. Now just imagine that your cell membrane is now very permeable at -5 degrees centigrade. You have high salt concentration here. The salts enter the cell and they generate havoc inside the cell. So that's one mechanism of cell damage. And you can see here a progression of what's happening with red blood cells. As you go to lower and lower temperatures, actually the water leaves the cell because the extracellular composition is higher, and the chemical potential inside the cell is higher than outside the cell, and the water leaves to equilibrate the difference in chemical potential. So mass transfer across the cell is the first mechanism of damage to cells during freezing. Now, chemical damage is temperature dependent, and it has a component of time. If you have the ability to bypass this region in which there is chemical damage very fast to very low temperatures, then you reduce the chemical damage. And amazingly enough, cells die not from the ice, but the fact that the ice has rejected salt, and they suddenly find themselves in a high concentration salt solution. So the first part of the curve is due to the high concentration of salt, and it is possible to develop mathematical models that describe what's happening when a cell finds itself inside a region in which there is ice outside the cell and high concentration of brine around the cell. The cell essentially will start to shrink. And again, I will not go through all the mathematical derivation, but this is typical to what's happening to the cell. As you freeze the cell with increasing cooling rates, okay, there is mass transfer, and if you go to low temperatures faster, there will be more mass, more water inside the cell at any temperature, because it's time dependent, okay? So at 1,000 degrees centigrade per minute, here you have almost no time for the water to leave the cell as you go to very low temperatures. On the other hand, if you go to 1 degree centigrade per minute, most of the water will leave the cell will shrink by the time you get to -20 degrees centigrade. The shrinkage of the cell means that the concentration of salt inside the cell has become very high. So you have chemical damage, and obviously the chemical damage is related to the cooling rate. And as you go to higher cooling rates, you will have less chemical damage at any temperature. So let me just show you some experimental results. This is how cells look when they are entrapped between the ice crystals. You can see that all that is left from them is just a ghost, just the cell membrane. They're all dead, but perhaps the most interesting thing is that they're all along the ice crystals. So that's where they find themselves. And this brings us to the effect of low cooling rate. So when you freeze something with low cooling rate, the survival increases as you increase the cooling rate, because you get to lower temperatures before mass transfer damage has time to occur. Now, what's happening when you get to this point? When you get to this point, you suddenly detect intracellular ice form. And this is an early study, I think, beginning of the century, that monitored what is happening inside the cell when you freeze it, let's say, fast. The water has no time to leave the cell. So it's inside the cell. But the concentration of the cell inside the cell is low, because the water didn't have time to leave. The first part of the survival curve, namely, as you increase the cooling rates, the survival increases, because the concentration of solids inside the cell is at any temperature is low. The consequence of that is that the cells become supercooled. And if the cells become supercooled, then they can freeze intracellularly by sudden nucleation. And we have done some experiments looking at intracellular ice nucleation as a function of the cooling rate. And what we found is, of course, one thing that happens is that when ice freezes, optically it stops being, the water stops being transparent and appears dark. Obviously, with X-ray, you can also see that. So what we found is essentially that there is a correlation between the rate of cooling and the supercooling and the formation of ice. The higher the cooling rate, the greater the supercooling, the more chances for ice formation, and the cells die from the second mechanism, which occurs at high cooling rates, which is related to intracellular ice formation. So in total, there are two mechanisms of damage. One is at low cooling rates, which is chemical damage. And it decreases with an increase in temperature. And the second is mechanical damage due to intracellular ice formation. And the damage increases, the survival decreases with an increase in cooling rate. Therefore, there is an optimal cooling rate for each type of cell. The issue is that this optimal cooling rate is not the same for all cells. It depends on the cell membrane permeability, on the cell membrane dimension. You can see here, for yeast, optimal is 10 degrees centigrade per minute. For red blood cells, it's 3,000 degrees centigrade per minute. So this is essentially one of the main problems. Different cells require different optimal temperatures. Now, this is for those of you who are essentially biologists in art. And there are additional mechanisms of damages in addition. So this is the work from John Baust. And essentially, there is apoptosis. Cold is a trigger. It's an input. And it can affect various molecular pathways. And cells can decide whether they want to die and undergo apoptosis. And there are chemical additives that reduce the chances for apoptosis or inhibit apoptosis. And there are other chemical triggers that are being used, which I will not discuss today, but they are part in designing the solution for preservation. So now the principles of cryopreservation. As I mentioned earlier, in 1949 it was found that the addition of glycerol improves the survival of cells to minus 79 degrees centigrade. So experiments show the following. They show that as you increase the concentration of those chemicals known as cryoprotectants, you improve survival, and the survival is improved during freezing with low cooling rates. Obviously, what this means is that the effect is on chemical concentration of the solution inside the cell. Why does it happen? Well, the cryoprotectants enter the cell, most of them. They replace the water in the cell, and therefore they dilute the concentration of the salts because now you have glycerol inside the cell and you have glycerol outside the cell. You've replaced part of the water by glycerol, and now you don't have any more, when there is mass transfer, you don't have any more high concentration of solids. And so the mechanism of cryoprotection occurs with low cooling rates. This is the region in which cryoprotection works. And again, different cells have, again, different optimal temperatures. And this is how freezing occurs in the presence of glycerol. You've seen earlier how freezing occurs without glycerol. You can see that the structure of the ice crystal is changed, but perhaps this is the most interesting observation. This is how cells look after, red blood cells look after freezing without glycerol. See only the ghost, and they are along the ice crystals. This is how they look after thawing with glycerol. So there are cryoprotectants. These cryoprotectants modify the composition at the interior of the cell, reduce chemical damage due to increase in salinity and concentration, and essentially that's how survival is being achieved. So it's also important to realize that cell survival is cell type dependent. So you don't have the same cryopreservation protocol for an embryo as you have for a sperm, because the mass transfer is different, the dimensions are different, and that's important to realize because I'll discuss that soon. So one of the issues is how to introduce the cryoprotectants into cells. How do you get them into the cells? Of course, they have to go through perfusion. You introduce the cells in a solution. Glycerol, most cryoprotectants are permeable. They can enter the cell, you generate an equilibrium. And actually, again, there are mathematical models for the introduction of cells, in glycerol in a cell. I'm just mentioning that so that you know that there are models for that. And these are the results. When you introduce a cell in a solution of glycerol, you first see that the radius of the cell goes down. Can you figure out why? Because the permeability of the cell membrane to water is much higher than the permeability to glycerol. So to equilibrate the difference in chemical potential, the water will leave first. Okay? But then glycerol says, well, I want to go in because the concentration of glycerol inside is low. So the glycerol starts moving in, and then you get back to the normal dimension. So it's a very interesting phenomena of mass transfer. So this field has quite a bit of mathematical modeling.
What's the order of the time?
Of the time? I don't know right now, but it's minutes at least. It is not instantaneous. The mass transfer is a time-dependent process. So cryopreservation of cells can be achieved. The mechanism is pretty much understood. As you know, we're using freezing to cryopreserve pretty much cells in pretty much every laboratory. And the cryopreservation protocols are cell type dependent. So this is one mechanism to preserve cells. Another mechanism to preserve cells is vitrification. Vitrification is a process in which the solution viscosity is increased to values in which the fluidity of this glass is so low that any perceptible changes do not occur in the time scale of our life. And you can see that with glasses. If you look at glasses at old churches, you can see that there is a deformation of the glasses because the glass, the windows, are essentially very viscous fluid. And of course, in our lifespan, we don't notice the changes in fluids. So why is viscosity a possible solution to cryopreservation? Because if you completely fix the water molecules in place, then there will be no changes. Okay? You can preserve them forever, or in our lifetime scale, forever, without any chemical changes, anything to happen. How is this done? It is done by increasing the viscosity of the fluid. How do you increase the viscosity? Again, with cryoprotectants. You introduce sufficient high viscosity cryoprotectants so that the water, as you lower the temperature, the water becomes into a glass. Now, this process is strongly dependent on cooling rates. So, from a thermodynamic point of view, you have several possibilities. One is, and again, vitrification in biological materials depends on the concentration of the cryoprotectants. If you replace completely the water with a cryoprotectant, then obviously there will be no ice crystal formation. So if you eliminate it. There are three domains. One, which is unstable vitrification. In the unstable vitrification, the chances for ice formation are very high. So you can see it as this is a plot as a function of concentration. If you have this level of concentration, in order to increase the viscosity sufficiently, you have to get to very low temperatures extremely fast to capture the water molecules in place. For this, you need thousands of degrees centigrade per minute. Now, heat transfer is a time-dependent process. You've seen the equations for heat transfer for phase transformation. So you can really do it with only extremely small volumes. And that's one area of research in which people work on vitrification with very small volumes. Then you have this metastable state. And in the metastable state, the water in the solution ends up being supercooled, like thermodynamically supercooled. But if you go fast enough so that ice crystals do not form, then again, at low temperatures, this is the so-called glass formation curve. This is the temperature at which glass formation occurs at different concentrations. So if you go very fast here, then you will not have ice crystals, even though it's a metastable state. And then this concentration is essentially a concentration in which there is always vitrification, no matter what the cooling rate. So here you can go extremely slowly and doesn't matter, and you'll still have vitrification. So these are the domains. Now, vitrification is a function of only the composition. It is not a function of the cell type. And I'll show you shortly, this is an important observation. So this is a typical curve. It shows you that for different cooling rates and composition, there is a path in time that will cause vitrification. And if you go with this cooling rate, then you'll have ice formation. And that's the whole field of vitrification, namely what composition and what cooling rates cause vitrification. And vitrification is good for preservation because once you have vitrified something, nothing can happen. Chemical reactions cannot happen anymore. Now, the first work on vitrification is a paper by Luyet in 1938, and I would say the most important modern work in vitrification is the work by Greg Fahey and Bill Rao, who preserved mouse embryos. Right now, cryopreservation of embryos and of oocytes and of sperm really gives hope to so many people that would not be able to have children. And so this field has a tremendous impact on life as we know it. And both of them are being used. There's another element to vitrification, namely the warming rates. If you're in the metastable state, and if you have pockets of fluid that went very fast, the glass formation, and were in a metastable state, because you went very fast to the glass temperature, once you start warming them up, these pockets of ice, of water, can form ice because they're metastable. So warming rates are very important, and actually they have to be much faster than the cooling rates to avoid the formation of ice in those metastable pockets inside the solution. I've sent many of you the book on cryopreservation. It's a good book. And again, Greg Fahey is kind of the guru of vitrification, so his chapter 2 is worth reading. So just in summary, on oocyte, there are two methods for preservation. One is slow equilibrium freezing, the other one is vitrification. With slow freezing, again, water leaves the cell. You will look for an optimal cooling rate. With vitrification, essentially ice formation is avoided, and you need high concentration of solutes and high cooling rates. So these are the two mechanisms. Warming rates are also important. And in summary, cryopreservation of cells is pretty much understood. Now, how about tissue? Because we started this work by suggesting that what we want to do is to preserve a whole organ. So why can we preserve cells, but we cannot preserve organs? Because it should be obvious to translate what we need with cells to organs. Well, first of all, different phenomena occur when you freeze tissue or an organ relative to the phenomena that occur when you freeze a cell. The main difference is geometrical. In a cell, when you freeze a cell, the cell is completely surrounded by a solution. When you freeze an organ, the freezing actually occurs through the blood vessels, and the cells surround the ice. In cells, ice surrounds the cell in organs, cells surround the ice that goes through the blood vessels. So these are some electron microscopy work that I've done with Charles Lee. And you can see that when you freeze cells, actually ice forms through the blood vessels, but and the cells shrink. But as a consequence of the cells shrinking, the blood vessels expand, right? Because the water goes into the blood vessels. And one of the main mechanisms of damage is this expansion of the blood vessel. We have developed some models for the process of freezing in blood vessels using something known as the Croc-cylinder model. Croc-cylinder model is known since the beginning of the 20th century to model mass transfer. It's heavily used in pharmaceuticals. And I will not go to the model, but again, I can provide you with the details. But the most important thing is the comparison, what's happening to cells and what's happening to organs. So essentially, the tissue is the dotted line, the cells in tissue is the dotted line, and the straight line is a cell, a hepatocyte. And what you can see is when you freeze slowly with different cooling rates, pretty much a similar phenomenon occurs with the cells in tissue and the cells in the organ, the cells in the suspension. The cells shrink, pretty much similar for the same cooling rate. So this is one thing. Now this is intracellular concentration. Again, you can see that as you increase the cooling rate, the intracellular concentration at each temperature, okay, is lower. The higher the cooling rate, the lower the concentration. And again, cells in tissue and cells in a solution behave the same way. Supercooling is the same, and this is the parameters for viability of hepatocytes, they are the same. And what's the main difference? The main difference is the geometry. When a cell freezes and water leaves the cell begins to shrink. But the shrinkage of the cell is only about 0.8%, just because of geometry. Now, on the other hand, the blood vessels, if the cells shrink, the blood vessels expand. They can expand by a factor of 2. So this is part of a mechanical damage that occurs to the important structure of the tissue, which is blood vessels. So cells may survive, but the blood vessels do not survive. And therefore the cell cannot recover. So that's one mechanism. There are other problems, and that's mass transfer. For introduction of cryoprotectants into the cells. If you have a cell and you put it in a solution of cryoprotectants, then the mass transfer is relatively easy. It's around the cell membrane. To introduce cryoprotectants of cells in the organ, you have to perfuse the blood vessels with the cells. This is a much more difficult problem because the fluid flow is very slow. You have laminar flow, but extremely slow, particularly through the capillaries, sinusoids in the liver. So we've developed some mathematical models for that, and we find that physically it's impossible to actually introduce or at least the level of cryoprotectants that you want into the tissue, and that's a main problem. So again, for the interest of time, I will show you one more problem with freezing of organs, and that's thermal stresses. Organs are large. There is a difference in temperature when you freeze them between the exterior and interior. and these cause thermal stresses, which actually can lead to cracks. So the problem with organs is mainly actually several levels of problems. One of them is that cell tissues are made with different cells. The optimal cryopreservation of cells is different, of different cells is different. So whereas each individual cell can be cryopreserved with their optimal cooling rates, cryopreservation of whole organs, you have to adjust the different cooling rates. Hepatocytes will have a different cooling rate than endothelial cells. So you cannot fit it exactly. The second thing is the introduction of the cryoprotectants. The cryoprotectants, high concentration, are toxic. So we're pretty much stuck. At this stage, there is no successful cryopreservation of an organ. And you can realize the problems. The problems is that an organ is not a cell. An organ is made of many cells, but in a different configuration. Now, there is an interest in vitrification of organs, and so far, John Bishop has developed a technology for warming organs rapidly with nanoparticles, and that apparently is successful with a kidney which he transplanted, but again, it's a mouse kidney, and there is promise, but so vitrification is really considered the potential method. Why? Because it is not cell-dependent. Okay, cryopreservation with freezing, you have a variety of cells, and the survival is cell-dependent. You have to fit everything. Vitrification is only composition-dependent. It's independent on cells. So with vitrification, if you find a way to vitrify, then you don't care what cells are in that organ, because it's only composition-dependent. Some research was done with vitrification. It is there. I mean, it is considered that this is probably the way to go. Vitrification. The problem with vitrification is that you have to introduce relatively high concentration of cryoprotectants. You have to introduce various cooling rates, to control various cooling rates, warming rates. I think that the nanoparticles may have resolved the problem with warming rates, but it's essentially at the early stage of the field, but it has promise. So what are we doing in our group? We are working on isochoric supercooling and on 3D cryoprinting in order to overcome the problems that we have discussed earlier. So in our system, isochoric cryopreservation is done in a closed system, as opposed to isobaric, which is conventional cryopreservation, which is done on the atmosphere at constant pressure. So we've changed the thermodynamics from constant pressure to constant volume. And this affects pretty much everything that happens, but just to give you a sense, isobaric constant pressure occurs in an open glass of water. Isochoric occurs in a glass of water that is tightly sealed. Okay, we're trying to minimize air actually in that system. So we have three different technologies for isochoric preservation. One is supercooling, the other one is isochoric freezing, and the third one is isochoric vitrification. And I will describe to you what we're doing and why it may work better. So in isobaric freezing, you lower the temperature and all the water forms ice. In isochoric systems, as you lower the temperature, when ice forms, it essentially, because ice, in a simplistic way, ice has a lower density than water, it increases the pressure. Now, if you look at the phase diagram of temperature pressure, as you increase the pressure, you go along this liquidus line. Okay? So, as you lower the temperature, ice forms, but also pressure increases, and the system finds itself into a two-phase system in thermodynamic equilibrium. The ice and the water are in thermodynamic equilibrium. So in isobaric, once you lower the temperature, everything freezes. In isochoric, as you lower the temperature, ice forms in part of the volume, but the ice is in thermodynamic equilibrium with the water, but at a higher pressure. So you go down along this line, and if you were to keep your biological matter in the unfrozen part of the system, then it will not experience ice. It will experience an increase in pressure, but it will not experience ice. So you avoid the damage due to ice formation. And if you look at the percentage of ice at different temperatures, you can see that at minus 20 degrees centigrade, about 50% of the volume is unfrozen. So if you were to keep the cell in this volume that's unfrozen, it will not experience the mechanical effect, the effects of ice, and it will be in thermodynamic equilibrium because it's a system in thermodynamic equilibrium. So we are working on these three technologies: supercooling, freezing that I've just discussed, and vitrification. Now, the isochoric freezing can preserve biological matter we found for weeks, days, and weeks. So we can extend the preservation between days and weeks, and we have, again, good results. And again, this is a recent result that was just published in Communications, Nature, and with the preservation of corals, centimeter scale corals with the vitrification solution that again Brooke and Tony have worked on, and I guess made the news just 2 days ago or so. So it works, and I think that we have to focus on this, on how to affect the metastable state during vitrification and how to increase it. And Gal and Allah are working on this, and perhaps we're going to get some more information about what's happening inside the vitreous system. Last, cryoprinting, and this is Linnea and Leo's work so far. Cryoprinting has a different concept. We know that we can preserve cells by cryopreservation. So the idea is, now in printing, you take small volumes and you assemble them into a large organ. So the idea is, how about if when we deposit that small volume that contains cells, we will freeze that small volumes with the optimal cooling rate for the cells inside the cells on that volume. So as a consequence, at the end of the 3D cryoprinting process, we will have a large organ that is completely frozen, but in which each element is frozen with the optimal cooling rate and with the optimal composition and so on. So by the way, there was a joke when I did my PhD at MIT saying there is no problem to freeze, to cryopreserve an organ. You just cut it in small pieces, freeze each particle separately, and then the only problem you have is to put it together. So now we have a way to put the organ together, and this is the general concept. And Gideon Ukpai has done his PhD on that, and Linnea and Leo are about to finish their PhD on this. But essentially what we do is we immerse the organ into a solution, a freezing solution, and we print each separate layer. And we freeze each separate layer as it's being printed. And as a consequence, we can also control the microstructure. And we have different technologies here. This is conventional printing, and this is freezing as you print. And this is a robotic system that actually can take different layers and assemble them into an organ. And this is cylindrical coordinates freezing. So recent results, this is the work by Leo, who has done, has 3D printed food for people with dysphagia by controlling the structure, the microstructure. And this is our most recent and to me the most impressive result. This is the work of Linnea, and she actually has developed a two-step freezing protocol, which is known actually, in which she freezes to a certain, with a certain cooling rate to a certain temperature, and then immerses the frozen organ into a freezer at minus 80 degrees centigrade. It can stay there. And then you take it out from the freezer, you thaw it, and you have a living tissue. And this is essentially the result. Her cells survived, and actually she managed to freeze 5 layers of tissue with the cells in each layer surviving the process. And some of the capstone students have actually gone through how many, 10 layers? Or?
I think 11.
11 layers. Okay, so right now with this technology, we pretty much have no limit to the dimension of the organ that we can freeze. So it has great possibilities. Now I want to finish with what nature is doing, because nature actually knows how to protect their organisms. They have evolved to protect. And there's 2 very interesting mechanisms. One is essentially the frog, the Canadian frog that can come back to life. And what we have done with Ken's stories, we looked at that, and this is another technology with antifreeze proteins. It turns out that fish in Antarctic, the Arctic, produce certain proteins that reduce the freezing temperature, but in a non-colligative way, without increasing the concentration. And these are antifreeze proteins, and together with Amira Rav, we actually discovered that they have another property. Namely, they protect, we believe, the cell membrane. If you remember, the cell membrane is a major site of damage. So if you find something that can coat the cell membrane and affect from the exterior the mass transfer across the cell membrane, then you can also increase the viability. And we think that that's what the cell membrane does. Now, with respect to the frog that survives freezing, again, work done with Ken's story. And what we did an MRI of the frog as it freezes. And dark is ice. Light means living tissue. I cannot slow it down, but if you look a lot, you'll see that freezing begins from the leg. This is the head, and this is the liver, this is the heart. Freezing begins from the leg, and essentially the frog gets scared, begins to produce glucose as a means of cryoprotecting, and essentially as the frog freezes, it increases the concentration of the cryoprotectants systemically, and the frog can survive up to about -5, -6 degrees centigrade and come back to life. Again, we have a couple of papers on that. We actually managed to use this combination of antifreeze proteins and glycerol, and we preserved hearts and livers and transplanted them. And I believe that there is lots of things to do in this field, and I hope that you will continue working in this field, because it has lots of exciting things for everybody. Molecular biology and heat transfer and mass transfer and devices. And I want to finish by saying thank you to Professor Crevallo at MIT. He passed away, but I owe him my career. Thank you.